Welcome to the first in this set of lectures, talks on sensory physiology. This one will be a separate one. I'll push through it as quickly as possible. But the subsequent one on this special sensory systems, very likely I'll split into smaller segments. So I don't want to say exactly how many there will be at this time. Expect at least 3, possibly 4, covering this topic. And remember that in this study of with an integrated approach to human physiology, future presentations, such as with the somatic motor system, will call our attention back to more detailed studies of some of the sensory circuitry in detail. So it'll be introduced at this time and then integrated into a more global systemic approach as we proceed. So about this chapter then, we'll talk about the general properties of sensory systems, the somatic senses, and then the one that will be split up, very likely, was intended as to be, this chapter was intended to be 2 presentations, but in looking at it more closely, it is going to take at least 3 and maybe split it into 4 so that you can watch these separately and not have to commit to such a gigantic block of time. So the general properties then, starting first with the sensory division. Remember, we're talking about both the somatic and the visceral sensory nervous system in general in this chapter, and we'll specify in particular cases when we are distinguishing between them. And certainly here we are considering the somatic senses, the somatic sensory system, and the special senses fall into that category of vision, hearing, taste, smell, and equilibrium. Do you remember which cranial nerves these are? Cranial nerve 1, cranial nerve 8, taste is going to be 7, 9, and 10. Smell is cranial nerve 1, and then the equilibrium again is cranial nerve 8, the vestibulocochlear nerve, cranial nerve 8. So in a previous lecture you were instructed to look at the table listing the cranial nerves and to study that material and come to know it, and now associate it with particular functions. Now for the typical spinal nerves, or in some of the cranial nerves that carry these types of somatic senses, such as cranial nerve 5 that will carry sensory information from the face and much of the head, and the proprioceptive type of information from the muscles of mastication. So when you are chewing and eating, moving your tongue around in your mouth and things like that, that's carried as a somatic sense within one of the cranial nerves, the cranial nerve 5, that innervates those areas. So familiarizing yourself with the cranial nerves for both in the context of both the special senses and the general somatic senses of touch, temperature, pain, itch, and proprioception are important. So when you get a little itchy spot on the end of your nose, what cranial nerve is that being carried through? Information processing by the sensory division include that which is usually subconscious, such as the visceral stimuli that I said would be mentioned, and some of the somatic stimuli, such as the muscle length and tension. So you are able to chew and manipulate food in your oral cavity without having to give particular attention to exactly the moment-to-moment changes in the positioning of the tongue. So it is not without conscious awareness or the capability of focusing attention to sense it, but it's really automatic. So that's kind of what's being inferred here. And the proprioception, as I had mentioned. So remember, the tongue is made up of skeletal muscle, as are the muscles of mastication for chewing. So I use that as a prime example for the subconscious type. But in any muscle that you have, there are signals being sent and the degree of stretch and things like that. When you do the tendon stretch reflex test, playing around, or clinically, there really isn't a conscious level of awareness of that information. Oh, my quadriceps muscle just stretched. I mean, you see your knee jerk and you go, wow, that's pretty cool. When you can place your hand on your quads and feel them contract, but you don't really feel them stretch. So these are the kinds of things we're talking about here. But we know that beyond this, some aspects of proprioception, you can tell when there is a torsion of your ankle, especially if you twist your ankle, then the pain becomes involved. Then it kind of exceeds this subconscious level that we're talking about here. So we're not trying to be completely exclusive. We're trying to categorize and within reason associate the subconscious with the conscious levels of awareness, and when one can move into the other realm. The visceral stimuli typically, as we know, will stay subconscious. So the distension of the gastrointestinal tract, unless it's going to produce a pain or an irritation of the nearby body wall, you're not really going to be able to tell that it's there or where it is, but that's not a 100% as you might know. You can have blockage of the gastrointestinal system and know about where it is in the gut tube. Quite often you can feel and even hear the gas crossing horizontally just below the rib cage in the front of your body as stuff moves through the transverse colon from the ascending colon on the right side to the descending on the left, or feel it drop down and go through your descending colon and into your sigmoid colon and rectum, and then you're on your way to the restroom, right? If you've ever had diarrhea, you might have felt that. So again, we're trying to remind ourselves of what exactly does stay subconscious, and that with some degree of accuracy and localization, and even a big degree of lack of inaccuracy of localization for the visceral stimuli. The blood glucose concentration, certainly we cannot feel that. Internal body temperature, it can cross over, as you know, to the level of conscious awareness, osmolarity of body fluids, until you are feeling a dry mouth and maybe dizzy and thirsty. You wouldn't know much about the osmolarity of your body fluid. Lung inflation, for the most part, unless there's pain, limitation, or pathology or extremes, you really never become aware of that. The pH of the cerebrospinal fluid, the pH and the oxygen content of the blood, of course. If that's related with you having a restricted ventilation, you can feel like you're suffocating, but it's not the same thing as sensing, perceiving those sensations directly. The sensory pathways, then, we want to think of a stimulus as a physical energy that will impact a sensory receptor, and the receptor acts as a transducer. It transduces that physical energy, a signal from the external or internal environment of a physical nature, and transduces it, as you know, into a neural signal that will have some meaning. So therefore, there's going to be a transduction to an intracellular signal, and this usually, of course, is a change in the membrane potential. And so this stimulus would have to reach a certain threshold such that, as we know, there'd be a graded potential that would lead to the triggering of action potential events excuse me, at the trigger zone. And then this will be carried to the CNS. So we'll look at this in more detail as we proceed, but in previous presentations, when we talked about the action potential, we used the primary sensory neuron, or the first order sensory neuron, as an example to talk about triggering action potentials at the trigger zone. And in that case, it's located far out on the peripheral process, and we'll see it again here. So we're not talking about an axon hillock, as you recall, where the trigger zone is in many of the other neurons. most of the other neurons. So the integration in the CNS, of course, is really ultimately at the cerebral cortex for conscious awareness. Even there can be subconscious perceptions that lead to actions. All the subconscious actions do not have to be restricted to triggers within the brainstem, the lowest levels of the brain. In some situations, probably very few, triggers for an output might occur in the brainstem, and simultaneously and rapidly the information is shared with the cerebral cortex. But even in a situation like that, the cerebral cortex would have been recruited in the preparatory stages for making such a decision. You know, I use that term a lot, but as you know, there has to be some final event of integration to determine a patterned outcome or output of the nervous system. So to reduce it down first to just this single neuron, it is a pseudo-unipolar neuron with a peripheral process going out to the periphery where the receptor will exist. In this case, the receptor mechanism is within the free nerve endings. And I draw your attention in this picture of the telodendria type of a spreading and distribution of the terminal portion of the distal process as well as the central process. They're going to turn those into just single little structures for more reduced illustrative purposes as we proceed. And I'm going to remind you that it looks like this when we get there. So when they show the illustration of the receptive field for one sensory neuron, remember that it would encompass this area, and that's where all these processes spread out to innervate that portion of the receptive field. They're just going to show one little dot at the end of it, and then a circle that it covers. Well, that's because these things spread out within that circular area. Anyway, I would prefer they did not reduce the detail in their illustrations, but they have. Well, this we see the peripheral process is unmyelinated. Of course, it can be myelinated, but if it's unmyelinated, the conduction is going to be slower. So remember, the trigger zone is somewhere out here distally on the peripheral process. So we're talking about integration of information between the site of reception, the initial membrane event that collectively will converge on the trigger zone and integrate into either subthreshold or suprathreshold or just barely threshold signals, right? And then that'll determine the nature of the neural code passing in the peripheral process and then bypassing the cell body to the central process and being delivered to the neuron population within the CNS. So here we are seeing an example of a myelinated peripheral process as well as a myelinated central process. So this is going to be a faster conducting organization, structural organization. The other difference in this illustration is that it's a more complex somatosensory receptor in that the single terminal expansion, so it's not a telodendria, it's a single terminal type of expansion, is associated with other tissue entirely, and so the nerve ending is completely enclosed in these layers of connective tissue. You may recognize this as the proscenium corpuscle, but there's slight modifications on this. So this would be sometimes it's called a receptor organ or a receptor apparatus, a receptor structure, various terms used for it, and that's to indicate that there is something in addition to the nerve ending required to transduce the signals generate the stimuli for signal transduction into the primary sensory neuron, or the first order sensory neuron is the other term that we'll use for it, and I'll probably say both every time. Here again we note the myelination, both peripheral and central processes. However, the distal portion, or the terminal portion of the peripheral process in this case is associated with a unique and completely separate type of epithelial cell, in this case a special sensory receptor. And this one, as you recognize, is a hair cell and can read. And what these receptor cells themselves actually do is transduce some type of a signal. In this case, of course, it's going to be mechanical, the deflection of these hairs, the cilia, on the apical membrane, and transduce that into a membrane and an intracellular signal to cause exocytosis at the base of the cell. And that then gives us this appearance of a typical type of chemical synapse. However, it's not an interneuronal one. It's a specialized one between a receptor cell and the peripheral process, the dendrite, if you want to think of it that way, receiving synaptic input of the primary sensory neuron, the first order sensory neuron. This lets us then transition to a brief discussion of the types of sensory receptors, the type on the left column, and examples of the stimuli that they would receive receive. Let's just leave it at that, not throw in the term perceive here just yet. So the chemoreceptors, the mechanoreceptors, the photoreceptors, and the thermoreceptors. So quite obviously from their names you have a general understanding of what they're going to be capable of reacting to as stimuli. So oxygen, pH, the hydrogen ion concentration, various organic molecules such as glucose. These are very important elements within cells of sensing systems. There's even a region of the brainstem right at the base of the fourth ventricle, just where it enters the central canal. It's called the area postrema. That's the emetic center. So if something from your intestines triggers the release of molecules into the blood that are going to cause you to vomit, this is one of the areas of the brain that's highly sensitive to the molecules in the blood. So the chemoextensors extend beyond simply what we see here. The mechanoreceptors, responsive to pressure, they're called baroreceptors. The cell stretch, the osmoreceptors, that's kind of an odd thing. Cell stretch, yeah, because the cell either swells or shrinks. Threw me on that one. Yeah, so it's cell stretch, not like a muscle stretch. Osmoreceptor would be just one example of this. The stretch of neuron endings around skeletal muscle fibers, you might recall the spindle apparatus, and certainly we'll study it later, is also a cell stretch, but it's the stretch is transferred to the nerve endings around the modified skeletal muscle cells, and that also triggers it. It's not an osmoreceptor event when you stretch your muscle and it triggers the proprioceptor response. So vibration, acceleration, and sound are some other examples. And then photoreceptors, obviously the photons of light, and then varying degrees of heat. And you know that heat can alter the structure, the chemical structure of proteins. And so these receptor proteins on the cellular membranes of cells, cytoplasmic membranes of cells, actually change their conformation with temperature, and that's how they transduce the signal to impact the receptor cell. The stimulus energy, therefore, is converted into information that is processed by the CNS, so that is sensory transduction. The ion channels, or second messengers that are altered, initiate the membrane potential change. And remember, if we don't get a neurophysiological response, in other words, a change in the coded message, up to this point I have stated it is frequency modulation and the interpulse interval time span. And for each of these there can be a duration. So in prior lectures I have stated you get a pulse of action potentials at a certain frequency for a period of time. Well, that's going to be the duration. And then obviously we have a temporal duration of the interpulse interval. So I want to bring that to your attention at this point. We'll talk about duration in a little while. But remember that the only signal, codified signal, that is of any value to the nervous system is the number of action potentials that are fired, how long that continues, and then how long in between the end of that burst and the next burst that's coming. I mean, there's obviously, how many bursts there are and stuff like that. It goes on a bit more beyond that, but I think you get the basic message there. So the adequate stimulus then is that which is the preferred form of stimulus for a particular receptor system. So is it adequate to cause a codified signal within this particular signaling pathway that's going to carry a certain type of information that has a certain perceptual meaning to the nervous system, and then that reaches the level of conscious awareness and is of value to the organism or perceived by the organism. So the threshold is a minimum stimulus, so the adequate stimulus is the preferred, the threshold is the minimum, and then the receptor potential is the graded potential. It's the change in the sensory receptor membrane potential. So it is actually a graded potential, and they can summate, as you very likely know. So the receptive field of sensory neurons is what we are considering in this slide, and this is where I try to draw that image back into your mind of the telodendria, the branching of this distal portion of the peripheral process, such that it would cover this entire highlighted area in this color. Same for this one in this color, and same for this one in this color. So it doesn't just end here, and then it's capable of picking up a signal from way out here. If it there is a certain distance away from it that it can pick things up, but the way it covers a rather a receptive field of dimension, recognizable dimension, meaningful dimension, is through the branching of the terminal process here. And then again, we see them converging onto a single neuron in the spinal cord, and we don't get these single terminals. There is the telodendria there, either onto one neuron or onto populations of neurons, but for the sake of simplifying the circuitry to understand this, we're seeing it as all three of these converging with a single synapse each onto a single target neuron. But remember, there's populations of neurons in here, but it's still the same essential convergence onto that population of neurons. And then those neurons are going to contribute to the information either integrated locally, but in particular that would be sent ascending up to the higher levels. Now, I can tell you specifically that this illustration is very likely to be representing a somatic pain and temperature pathway, because I know that synapse is in the spinal cord with the second order sensory neuron, and we have the crossing, the decussation of the pain and temperature pathway in the ventral gray commissure, as it's called here, and the information passes, ascends in the contralateral white matter. Another name for the pain and temperature pathway is the anterolateral system. So there's other cell populations in this pathway where this axon crosses and it ends up in part of the anterior white matter, the ventral funiculus here, or the anterior funiculus, one side and then the other side, excluding the gray, of course, and then part in the lateral funiculus. So we had talked about that with the anatomy of the spinal cord. So I don't want this to be misleading and misrepresenting at this point. For the touch at the tip of your finger, the neuron is not going to be here. These are going to turn and ascend into this, the dorsal white columns, as they're called here, up to a higher level in the brainstem, and we'll see that as we proceed. And then, so if you take this neuron and just move it up to the medulla oblongata, then it would be the same thing, just at a higher level. Okay, so again, they've simplified here to make a point, and I want to distinguish it at this time. So we're using this illustratively to focus on these three receptive fields that do have some physical overlap, as do the telodendria-like processes, terminal processes of the distal ends of the peripheral processes of the primary sensory neurons. And so how is that represented in the CNS is the other part of it. What we are looking at here then is the skin surface, and you see that they're making contact with the skin using this compass, separated, the tips separated by about 20 millimeters. So we're not pushing the pins to cause a pinprick type of a pain. It's just a light touch or light pressure. So that means this second order sensory neuron is going to be up in the medulla oblongata, not in the spinal cord as they had shown in the previous slide. So you see the receptive field for each of these. There is overlap, and the two that are contacted are shown in the green. So we see the two neurons that are going to fire the fastest from this signal. Nearby ones might be activated a little bit, but they won't be the fastest. So picture here, they're showing the telodendria type thing. So picture this as your hand with your fingers up there, and you'd have to have more fingers, I guess, both hands with all your fingers in there. And when this is right in the center of your field covered by that hand, more and more of your fingers are going to be activated. But if it's just kind of at the edge, only a couple fingers might be activated. So the green neurons here are going to be most activated, and then the ones that border that and have some overlap might be activated a little bit. So it's important to keep this in mind. Still, this is the first way of viewing how 2 points can be resolved on the skin, and it's called 2-point discrimination. But it varies with the size of the secondary receptive field. So this is the primary receptive field that we had indicated here. So the receptive fields of the three primary sensory neurons overlap with one large secondary receptive field. So each has a primary receptive field, and then there's secondary receptive fields at the overlap. So here in the convergence in the CNS, so here we can see that these 2 points of contact all fell within the the collective secondary receptive field for all these first-order sensory neurons, or primary sensory neurons, that converge on the same or the same subset of second-order sensory neurons, and therefore it's perceived as a unique individual stimulus. In other words, if this was your back or areas of your arm and lower arm or back or lower extremity, then you would perceive that as one point of contact. Whereas at the tip of your finger, where it's where it is a one-to-one, one receptive field to one primary and one secondary sensory neuron, then you have a very high resolution of two-point discrimination. Certainly the 20 millimeter is easily distinguished. And so that's approximately the range. So think of your lips have a very close very high resolution primary receptive fields and other parts of your body as well. Typically the ventral surfaces of the body are the ones that have the more highly resolved two-point discrimination, a denser one-to-one population of primary receptive fields to primary sensory neurons to secondary sensory neurons than any of the structures on the dorsum of the body. So our face and the front of our neck and our chest and the soft parts of the front of our arms, the palms of our hands, the abdomen, and of course the genitals, right? So the integration by the CNS of this information, then, this sensory information, is via the spinal cord to the brain by ascending pathways, and of course it'll be direct to the brainstem via the cranial nerves carrying that type of information. The visceral reflexes are integrated in the brainstem or the spinal cord, and they usually do not reach the conscious perception, as we know. We'll talk about referred pain at the end of this talk. The perceptual threshold is the level of stimulus necessary to be aware of a particular sensation. And in the textbook, the authors give a nice brief description, an explanation of how the higher centers can have inhibitory circuitry to attenuate the perception at various levels, and for you to be distracted and things like that, or for us to resolve the background information. In other words, to refine signal-to-noise ratio type of a thing, right? So what's the important signal, and you inhibit the unimportant, or what you think is unimportant, and you heighten the important information, and you get a resolution of signal-to-noise ratios. So the sensory pathways, each major division of the brain processes one or more of the sensory types of the sensory information. So we saw this in the anatomy talks, and now we'll just kind of begin some of the circuitry here to introduce the idea. So to get oriented, we're obviously looking at a lateral surface of the left cerebral hemisphere, and we are seeing as well the brainstem and the cerebellum from the left view. Superimposed as if we are able to see right through into the diencephalon, we see the hypothalamus, the thalamus here as well. And one point we are going to make is that the thalamus, as we had stated before, is a major somatomotor sensory relay circuit, as well as for relaying emotions. But for now we're thinking of somatosensory transmission from the primary sensory neuron through a multisynaptic pathway ultimately to the cerebral cortex. So first then is the olfactory pathways from the nose projecting through the olfactory bulb to the olfactory cortex. Okay, I'm going to go to the pointer because I just cannot bear to leave it this way. This is absolutely incorrect. Actually, the brain sits a little bit farther forward, but anyway, the special sensory receptors for smell are up in this area here in the nose, and the olfactory nerves go straight into the olfactory bulb. So their anatomical illustration isn't the greatest. Coming from down here, these actually are going to be different types of receptors down in our nostrils. That's not where we smell. The smell is up very high in the nose and goes, actually it's so high up it just goes straight up. So the olfactory bulbs are sitting right here, and it goes straight up into the CNS. Anyway, I just couldn't resist that. I'm sorry. So now that I have the pen on, I will circle a few of the others. So the olfactory pathways go right into the olfactory bulb, and note the olfactory bulb then becomes what's called the olfactory tract, and then so the second order sensory neuron is in the olfactory bulb, and its pathway is the olfactory tract, and then the third order sensory neuron is already in the cortex. So this is a distinction for a major point to be made for all the other pathways. So most of the sensory pathways project through to the thalamus, and there is a synapse before that and there. And the thalamus modifies and relays the information to the cortical centers. So there's typically going to be the first order sensory neuron from the receptor, such as the taste buds in the tongue, into the CNS. Then there's going to be a synapse between the first and the second order sensory neuron, and the second order sensory neuron is going to send information to the thalamus. And then there's going to be another synapse. I hate it when it jumps across like that. And then the second order sensory neuron synapse is on the third order sensory neuron, and then the third order sensory neuron is going to project into the primary the primary sensory cortex for that modality. And of course, the one we just followed is the gustatory, here to the primary gustatory center that we had indicated in our previous talk. So for all the sensory pathways, excluding the olfactory, there will be a relay through the thalamus. The equilibrium pathways project primarily to the cerebellum. As you might recall, the cerebellum is important for our balance, as well as many other critical functions, somato and visceral, sensory motor integration. It's very critical, but if there's injury to the cerebellum, then balance is not very good in that person. Oh, I thought there was one more, sorry. But still, for the equilibrium and all the other pathways, the information is going to have the branching of a primary sensory neuron to a secondary sensory neuron to a tertiary sensory neuron, although you'll more commonly hear the first order sensory, second order sensory, and then the thalamocortical pathway is created by the third order sensory neuron. So we see equilibrium will reach some general areas in the somatosensory cortex to be integrated, and we see the sound complex pathway, not just the simple 3-neuron pathway, but parts of it are simple 3-neuron pathway. The first order, second order, third order from the thalamus to the primary auditory cortex that we saw right on the upper more posterior portion of the temporal lobe. And then the somatic senses will be following separately, but either the synapse between first and second order right in the spinal cord or in the medulla, and then the second order sensory neuron to the thalamus, and then the thalamocortical pathway to the primary somatosensory cortex for the third order sensory neuron. So that's really just previewing it for you, and we will specify each time we study a particular pathway exactly where the first, second, and third order sensory neurons are located to emphasize it at those times. The properties of the stimuli first then are the modality. So this would indicate be indicated by where the sensory neurons are activated and the neurons terminate in the brain. So if you took a sensory receptor that had a very particular type of a structural organization in the periphery, and you could find its axon terminals in the central nervous system, and shift it to innervate a different population of neurons there that are associated with, say, pain and temperature, then fine touch at the tip of the finger would be perceived centrally as pain and temperature, correct? So it's really the circuitry within which you find these neurons that we have the modality giving any meaning to the organism. But they are highly organized, and so it's rare that there's a shorting out of the circuitry such as that. And then that means that the specific type of receptor in the periphery will be associated with that type of perceived modality in the CNS. So the system is organized to have specialized receptors in the periphery to be responsive to an external stimulus, environmental input that has some meaning assigned to it by the central representation of that modality. The labeled line coding is a big part of that conceptualization that we just put together. So in other words, if a different type of stimulus activates that receptor, then the central perception, the central representation, can only be in that one particular modality. So one example that the authors give that I'll use for right now, you get punched in the eye and you see a flash of light. So the photoreceptors have not been activated by photons, but somehow their membranes have been depolarized. And so the only way that that is represented by perception in the CNS is as light. Right? Or if you implanted a stimulating electrode in the retina and you used electricity to activate them, centrally it could only be perceived as light. So that's what this labeled line coding means, is that you have a one-to-one association of the receptor with the sensation. If you stimulate a pain receptor by whatever mechanism, the only central perception for that can be pain. So the next properties of stimuli is the location. So it's according to which receptive fields are activated. Where are those receptive fields located? So again, if you had a sensory receptor that is innervating the tip of the finger, and you somehow could take those endings, those fibers, and somehow move them and relocate them to the bottom of your buttocks, when somebody touched that area of the bottom of your buttocks, your brain would say, oh, that's the tip of my finger. Right? So the brain would not know any different unless you could educate it over a period of time. Anyway, kind of a goofy example or way to try to present it, but that does make the point that if the receptive fields are located in the tip of the finger, then the central representation of that as far as location is also the tip of the finger. So in other words, there is a somatotopic organization of the central representation of these peripheral locations. So the auditory information is an exception. It's sensitive to different frequencies. So it's not a somatotopic organization. It's another kind of topic. I forgot the term for it. You're catching me too many times doing that, right? But it is the frequency. So the lateral inhibition is a key feature of location as well, in that there is a built-in mechanism for resolving and refining the resolution of the exact location. So now we're not saying, well, that's my finger, my index finger. We're saying that is right next, yes, right under my fingernail bed in the front center portion of my fingertip, right? So somebody sticks a pin right up under your fingernail of your index finger, you can state, you can resolve exactly where it is, and we'll see an illustration of that in a moment. And then there is population coding, so multiple receptors that would be functioning together from one area, resolving the location, but obviously each having their own modality as a central representation, but the same location. So I'm getting both pressure and pain, and it's hot, so I get the temperature at that pinpoint under my nail bed of the index finger. So the properties of the stimulus, again, the location. This can have associations with differential distance. Hello? Sorry, I slid the book on top of my switch there and turned off the microphone. So if there are differences in the distance of a source of sound on the right versus the left ear, then the signals coming from the left reach the brain first, and the brain uses that to orient to the space and time associations of the source. As you know, that's very important evolutionarily to the selection of specialized neural systems, and so we would expect that it is a very sophisticated system. We're not as good at using sound to localize structures in our environment, but as we know, bats and dolphins, for example, are extremely sophisticated and specialized in those abilities. So let's examine the lateral inhibition idea in this slide. So it enhances the contrast and makes a stimulus easier to perceive. So here we are only thinking of the very small diameter light touch sensation. So we're not going to be taking the pin to cause pain. And the reason for saying that is it makes this a better system to understand. But also, by using that modality, these second-order sensory neurons are going to be up in the medulla and not in the spinal cord. If it was a pinprick causing pain, then these would be in the level of the spinal cord, as I had indicated on that other slide. So this is an area where you have a pretty high resolution of the primary receptive fields to begin with, and so we can see that the very limited input is to the exact center of this field in this illustrative example, and so neuron B is going to fire the fastest, and so in other words the highest frequency and the longest duration of that frequency, and probably a short interpulse interval as well. So then that digital to analog signal conversion means there's more neurotransmitter dumped on this second order sensory neuron than on the ones at the adjacent areas where it would be their secondary receptive fields, and they will be bombarded with smaller amounts of the neurotransmitter, and they might send pulses for whatever duration and for whatever length of time collectively toward their third order sensory neuron, but there will be an axoaxonic inhibition by collaterals of the neuron in the primary receptive field. And so what is going to happen is that this will turn off the periphery of the input and amplify the excitation of the central portion of the receptive field. So if you were able to envision the frequency of the action potentials of the first order sensory neurons, they would be progressively higher frequency of action potentials the closer you would get to the actual point of contact to the stimulus. So the primary neuron response is proportional to the stimulus strength across the distance that would include these first order sensory neurons. And then the pathway closest to the stimulus inhibits the neighbors such that at the next level, the third order sensory neuron in the thalamus, you're going to get an enhanced resolution because you're going to get a decreased frequency of action potentials of the neurons surrounding that primary receptive field, but no real change in the frequency of the action potentials of the neuron involved in the primary receptive field. And so this exaggerates the difference between the two without having to amplify directly amplify the firing rate of the primary receptive field neuron. So at the level of the thalamus, you get a first stage of the enhanced resolution. Other properties of the stimulus would be the intensity, so that's coded by the number of receptors activated and the frequency of the action potentials. And then the duration, so it's coded by the duration of action potentials, and so that's in two ways. The frequency of a pulse over a period of time, that's duration, and then the duration over which you get a set of pulses with a certain interpulse interval. So this is we might say by durations. And then some receptors can adapt or cease to respond. So this is where we'll see that there is a change in the duration of the collective set of pulses with their interpulse intervals. And in this discussion here, we'll see both durations. So you can change one duration independent of another is the point. The tonic receptors versus the phasic receptors is what we will see on a certain slide. So tonic receptors are ones that you want to have the capability to fire at different ranges on a more enduring basis. So like blood pressure, you need to know what the blood pressure is. Your nervous system needs to know what the blood pressure is on a constant basis. The pressure receptors in the skin are going to be set up to be phasic, so where you know that you've experienced an event, and when the event becomes constant, then you don't need to receive that information anymore because it's not important for that particular phasic receptor to be telling you that the event has endured. There might be some type of tonic receptor there that would pick up the information and reflect the endurance of the sensory input. So the adaptation, the differences in the adaptation help to give meaning to the signal that's coming in to the CNS. So that, in other words, a little bit of perception that is real to be integrated into the perceptual capabilities of the central nervous system. So the sensory neurons use action potential, frequency, and duration to code stimulus intensity and duration. So here we are seeing a square wave input with a certain duration and a certain amplitude, correct? So that is a moderate stimulus. So the membrane potential here at the transduction site, that's going to be a graded potential, will decrease over time and distance to the trigger zone if it's super threshold. We talked about this before. Then it's going to lead to the firing of a certain number of action potentials that will, so that's the frequency, and this is just one pulse that we're looking at here, that will be conducted down the myelinated peripheral process, and the same number of action potentials, and it's a codified signal. So that will not change. Will proceed all the way to the terminal in the CNS, or the telodendria terminals for that first order sensory neuron in the CNS, leading to a certain amount of transmitter release. The longer and stronger stimulus will produce a membrane potential that will have a higher amplitude itself. You see that it's going up almost to zero, maybe like minus 5 millivolts, versus the minus 35, let's say, that we saw with the first amplitude, but also for a longer duration. So we see that the graded potential also continues for a longer duration of seconds here, nearly matching that duration of the stimulus. And so what we're going to get is a pulse of action potentials, and actually this is even changing. We get a change in frequency, probably stabilizes somewhere here early on, and then later begins to taper off. And this would be a single pulse, really, with a different frequency within it. And there may indeed be additional pulses of this similar pattern that would follow. So we don't want to think that there's just a single firing of one pulse of action potentials. There could possibly be an interpulse interval, and then the firing of another burst or pulse of action potentials. So the receptor potential strength and duration vary with the stimulus. The receptor potential is integrated at the trigger zone, and then the frequency of the action potentials is proportional to the stimulus intensity. Duration of a series of action potentials is proportional to the stimulus duration. So the neurotransmitter release varies with the pattern of action potentials arriving at the axon terminal. So now let's look at the tonic and phasic receptors and their adaptation at the level of the receptor portion, the transducing portion of the nerve terminal, and the events that occur at the trigger zone and what they're calling the axon of the sensory neuron, right? So this distal portion of the peripheral process where the trigger zone is located. Or even on the other side of the trigger zone to see what the action potentials would look like there. So the tonic receptors are slowly adapting receptors that respond for the duration of a stimulus. So they are going to produce action potentials over the entire duration. And this is just showing a change in the frequency. So the zero time is here, so this is start and this is end of the events there. So first there's going to be a higher receptor potential that will gradually fall off even though the stimulus intensity has not changed. And that falling off is going to be seen as a change in the distance between the action potentials. So these may indeed be bursts with interburst, or pulses with interpulse intervals, as we are seeing. So a 2 hertz with an interpulse interval, a short interpulse interval with a 1 hertz, with a longer interpulse interval with a 1 hertz, and so on and so forth. So there's coding within here beyond just the number of action potentials. But the important thing is that regardless of how we want to try to account for all of that, types of coding, we do have a coded signal being sent from the beginning to the end that is the same duration as the stimulus being applied. Then the phasic receptors will rapidly adapt to a constant stimulus. So here you see the entire duration of the stimulus over basically the same time as we saw on the previous slide, just kind of shifted to another location there, just physically on the slide of the picture. But the distance here is the same, so the duration is the same. And the receptor potential is just a single event that does not persist for the entire duration of the stimulus. And associated with that, which would be reflective of the amplitude of the stimulus, you get one burst. So the somatic senses, then, the modalities to describe, basically to state them collectively: the touch, the proprioception, the temperature, and the nociception. Nociception is inclusive of pain and the itch sensation. The proprioception, as stated before, is position in space, which is related to the amount of pressure, twist, torsion, and stretch within muscles and joints, and things like that. As we proceed through the various systems, we'll gain more insight into the subtleties of the definition of that term. Now we can take our first look at the somatic sensory pathways with the 2 locations of the second-order sensory neurons. Let me just check. Okay, this does go through the series here. And we recall that we'll revisit this as needed in the future, and so it is introductory. So for now, we remind ourselves the first-order sensory neurons are going to be associated with particular types of peripheral receptor structures, whether it's a free nerve ending or a Pacinian corpuscle, something like that. We will have a particular type of modality, or even the sense within that modality, the fine touch, proprioception, and vibration. And then the nociception, temperature, and coarse touch in this other set of receptors. And it's their central circuitry that really is different. So the the pain and temperature and the coarse touch cross the midline in the spinal cord, and they're only showing the second-order sensory neuron with an axon going into the lateral funiculus. Some also go into the ventral funiculus, and it's called the anterolateral pathway, pain and temperature, as well as the coarse touch. And then, of course, as stated before, for some of the primary sensory neurons, or first-order sensory neurons, such as with fine touch, proprioception, and vibration, the central process It may give off a collateral with some branches in here and synapsing locally in the dorsal gray horn, but the major collateral primary pathway for it is to pass into the dorsal columns, as they're called, of white matter and ascend all the way up to the medulla oblongata to synapse with the second order sensory neuron. And we're not adding in the names of these nuclei. That's fine. Then the next pathway that's formed up to the thalamus, you see the decussation, the sensory decussation here in the medulla, and a sensory decussation here in the spinal cord. This second order sensory pathway is called the medial lemniscus. So you may hear me calling this the dorsal column medial lemniscus pathway, is that for fine touch, proprioception, and vibration. And then the third order sensory neuron is located within the thalamus, and the thalamocortical pathway brings that information up to the primary somatosensory cortex, which, as you recall, was the postcentral gyrus. In both cases, you get crossing of the midline, the decussation of the second order sensory neurons, so they innervate the contralateral, the opposite side, thalamus, and so right body, let's see, this is an anterior view, it looks like, that we're seeing here. So this would be right hand side, this would be left hand side, so the right hand side of the body is represented on the left hand cortex. So the sensory pathways synapse in the thalamus, and the thalamocortical pathway brings it up to the level of perception in the primary somatic sensory cortex. And it's that primary somatosensory cortex that has the homunculus on it, the somatotopic organization. Actually, these pathways all the way up through have a somatotopic organization. It's important clinically if there's restricted damage, then you're just going to get one portion of the body losing sensation. If there's a lesion just in this area here versus right over here where the one is, versus this part of the cortex or that part of the cortex. So it's quite important. And you will notice that the sensory homunculus will be represented proportionally differently than the motor homunculus. So I mean, look at the index finger, the amount of cortex, primary somatosensory cortex dedicated to that, to the face, as I was telling you, and to the tongue, and to the hand in general. And then areas like the back and hips and the lower extremity don't get much representation at all. Genitals might look small there compared to other parts of the body, but they get a good representation of the cortex, as I said, being represented as a ventral part of the body in the palm of the hand and the ventral surface of the forearm. There is that high resolution. A lot of first order, second order, and third order, and fourth order, if you want to call it here in the cortex, sensory neurons dedicated to those particular regions. So you're more likely to find a single neuron here in the primary somatosensory cortex that would be activated by a single neuron originating out here in the tip of the index finger, correct? And here they're showing the thalamocortical pathway radiating up into there. This is called the corona radiata, a radiating crown of white matter going through the cortex from the thalamus on up. Well, let's go back to the skin and think of the types of receptors that we would find there. So the touch receptors in the skin are distributed either just in the deeper layers of the epidermis, or just immediately subjacent to the epidermis, or deeper in the dermis and the hypodermis. The hypodermis is shown down here with the fat cells. And then one other location, of course, and we'll start here and then might move our way around the slide like this. The free nerve endings associated with the base of hair follicles. And these will sense the movement of the hairs. The next that we would see, looking here just in the deeper parts of the epidermis, the first that we see other than some of these free nerve endings that are going to be shown over here, is where the end of the peripheral process is not a free nerve ending. It's associated with other tissue-like structures. The Merkel receptors, also called Merkel's discs, will sense steady pressure and the texture of structures. Then Meissner's corpuscle will be just immediately subjacent to the epidermis, and there is a receptor tissue associated with it, and it responds to flutter and stroking movements. And then we see the free nerve endings right in the epidermis, and these would be for the nociceptor system modality, and it will respond to noxious stimuli. So two examples of free nerve endings, those for nociceptors and those for sensing the movements of the hair follicles. And then we see a bit deeper in the dermis and occasionally in the hypodermis, these onion-like structures, these layered structures. We saw it earlier, the Pacinian corpuscle for vibration, as well as the Ruffini corpuscle that responds to the skin stretch. Those temperature receptors, the free nerve endings that we saw in the, they call it terminate in the subcutaneous layers, and that is not correct. The subcutaneous is way down in here. They actually terminate in the epidermal layer of the cutaneous tissue, and they are cold receptors, and they're lower than for lower than body temperature, and the warm receptors respond at temperatures above 45, and at the same time will trigger pain information. The nociceptors, as the free nerve endings, will respond to strong noxious stimulus that may damage tissue. They are modulated by local chemicals. So this brief statement in the slide says substance P is secreted by the primary sensory neurons, and that this mediates an inflammatory response and is associated with inflammatory pain. What it doesn't emphasize here is that this release is actually by the distal most terminal of the peripheral process. So the release is out here. See where these are located? Just in the lower portion of the epidermis and the first part of the dermis. So the substance P is released out in these areas and acts on target cells here to cause the release of histamine and other types of cytokines and things like that to trigger a local inflammation. So it doesn't get released in the spinal cord and cause an inflammation response in the spinal cord. This is released, it'd be like a dendritic release of a substance, and it's out at the site of the sensory receptors in the first place. So the nociceptor pathways are reflexive, protective responses, and trigger reflective protective responses, and they are integrated at the spinal cord, and obviously the withdrawal reflex is one we'll see when we look at it in greater detail with the somatic motor system. Excuse me, when we talk about the integration there. The ascending pathway to the cerebral cortex is what is the conscious level central representation for the sensations. For example, with the nociceptor pathways that we're considering here, would be the pain and the itch. And we know that we have the capabilities of distinguishing those perceptions. The somatosensory nerve fibers differ for the information that they're going to carry. And so here we see on this table the classes of the somatosensory nerve fibers. The type of fiber is shown here, then the characteristic, then the speed of conduction, then what it's associated with. So the A beta, or typically called just the beta fibers, are large and myelinated, and they have a high conduction speed of 30 to 70 meters per second. They actually have the highest on this table, and they are responding to mechanical stimuli. The A delta, or just the delta fiber type, are smaller, and they are still myelinated, but thereby, with the smaller diameter, they're going to have a slower conduction rate, about 12 to 30 meters a second, and they will be transmitting information about cold, fast pain, and mechanical stimuli. And the C-type fibers will be small and unmyelinated, and very slow, therefore, 0.5 to 2 meters per second, and that will be a system responsible for transmitting the cold pain signals, heat, cold, and mechanical stimuli. Continuing on with the pain and itch part of it, the pain is in part related to histamine activating the C fibers causing the itch, and so like poison ivy, if you get poison ivy or poison oak, it's actually an inflammatory immune response, and the histamine that's released from the local inflammatory response activates the C fibers causing an itch. Or a mosquito bite is another example, right? The pain will be subjective type of perception, and there can be a fast pain, sharp and localized, the pinprick by the A beta fibers, or a slow pain, kind of being hit with a steel pipe, will be more diffuse and by the C fibers, like a pressure, deep pressure, crushing type of a pain. So the gate control theory for pain control is shown in this series of slides. So we see the first order sensory neuron, the central process of a pseudounipolar neuron, and its collaterals, the telodendria that I keep reminding you of. When it enters the gray matter of the spinal cord, where it's going to terminate, here we're seeing the slow pain C fiber, so unmyelinated, and small diameter. It collateralizes, and in the unstimulated state, there is an inhibitory neuron in the gray matter of the spinal cord that has a basal activity and keeps the pain pathway turned off, so there's no pain signal being sent to the brain. When there is a painful stimulus, the C fiber is going to fire, and it is going to excite step on the gas pedal for the second order sensory neuron. As well, it's going to inhibit the inhibitory neuron, so it's going to disinhibit. It's going to take the foot off the brake. So it's doing two things, pushing the gas pedal, taking the foot off the brake by inhibiting the inhibition or disinhibiting this neuron. Other pathways converge on that inhibitory interneuron, such as the A beta fiber for touch or non-painful stimulus, and it is an excitatory input into the inhibition, the inhibitory interneuron. And remember I said that they will give off a collateral that will innervate some other interneurons in the spinal cord before going into the dorsal column. And this is one of the targets of those collaterals local in the spinal cord, the inhibitory interneuron to the second order sensory neuron of the painful stimulus pathway, the pain pathway, the anterolateral pathway. So this way you can, if you get a painful stimulus, you rub it in that area, and by activating this inhibitory interneuron, you make the pain go away, right? I don't really know. I don't think that's what's happening when mommy kisses a boo-boo, but we could think of that too. Clinically, of course, we do neither of those two. You don't go to the emergency room to get your boo-boo kissed by a mommy. But therapeutically, you might have prescribed TENS treatment, the transepidermal neurostimulation, and that's what that would be targeting. They'd set the certain frequencies and stimuli parameters for the therapeutic treatment such that it would be activating the gating mechanism to enhance the inhibition of the pain pathway. Well, we can make these circuits look very tight, very specific, but there's a lot of neurons and there's a lot of axons and axon collaterals in this gray matter. It's highly organized, highly structured. It's had evolutionary pressure on it, refining it over millions of years. But what it hasn't refined is the sensory input from the visceral organs and how that information is shared with the pain circuitry. And it is shared rather than dedicated to a separate pain portion of the pain circuit. So there is a shorting out, in a sense, in the somatic sensory pain system, where visceral pain receptors, extensive stretch or whatever it may be, toxic substances from a local infection or thing, so appendicitis will ultimately hurt somewhere from the toxins released by the bacteria, right? So it's this convergence of the visceral primary sensory neurons onto some of the somatic primary, along with, some of the somatic primary sensory neurons onto second-order sensory neurons or other interneuronal systems, that we get this shorting out of the visceral onto the somatic pain distributions within the body. So the second-order sensory neuron just carries that information up to the somatosensory cortex and says, you got pain around your belly button, and a good clinician will know that could be an early sign of appendicitis. So the pain is referred to different areas. Ischemia will be a pain that is caused by a lack of blood flow, and chronic pain is a pathological pain. We talked about a dorsal rhizotomy in a prior presentation on the anatomy of the nervous system to emphasize the point that you only have sensory fibers in the dorsal root. And of course there can be treatment with analgesic drugs such as aspirin, which is a non-steroidal NSAIDs, non-steroidal anti-inflammatory drug, but also an analgesic, and it inhibits prostaglandins and slows the transmission of pain to the site of injury.